Let's give all a big round of applause to John and welcome him to our seminar. Thank you for joining us today.
Thank you, Islam, for that great introduction. I totally forgot to take that out of my slides, which means that I'm probably going to start with telling everyone, hello, I'm John Baxter. So I guess this part of the presentation is always where your speaker tries to give some credibility as to why they're here in front of the camera. So I'd like to start off because I still like to think of myself as relatively close to being a student. So I'm going to start off with my PhD. I do have a background in mathematics. So a lot of what I do actually comes from that more sort of mathematical end. I love thinking about machine learning, but I always love thinking about it from a sort of deeper level, a more theoretical level, and not just what we can do with it. As Islam stated, I'm now an associate research professor at the LTSI, where I research into machine learning, neuroimaging, and human-computer interaction. And I'm also an amateur philosopher. Amateur, I love that word because in French it means that I love philosophy. I am a lover of philosophy, which is quite true, but that also means that everything in this presentation is simply an opinion. So if you want some good, hard mathematical facts, you'd be hard pressed to find them here. You can definitely get a lot of them from Islam and your other invited speakers. But my goal is really just to take this time to broaden the idea of what machine learning can do in the context of medical imaging, and at the same time give you a couple of things to keep in your head when you're trying to decide what problems to solve and how to solve them. So I'd like to start off with an analogy. So let's imagine that we have a patient, and this patient has cancer, and they're coming into the hospital for a routine follow-up. So they've received treatment before, they're coming in to talk to the doctor to see what sort of further treatment may be necessary, to check on the progress of the treatment that they're currently undergoing, or just to see how they're feeling. So when they come in for this checkup, they're probably going to receive some form of additional medical imaging, like a CT image. And of course, we all know that CT is three-dimensional X-rays. So it all starts off with acquiring a number of X-ray images, which are then reconstructed into a CT. Because we already know this person has cancer, we already know that there is a tumor in there, so we should probably also identify that tumor. If we want to know things about it, we should probably delineate it, that is, outline it in the image and measure it. That way we know things like how many tumors there are, if they've changed size, or if they changed geometry, like if they've become more spiculated or uneven looking. Now, because we've seen this patient before, we have their prior images. So we may want to perform something like image registration, that is aligning the images that we've just collected of the patient to some of their images in the past. And this would give us some idea of if there are other changes that have been happening in this patient. If you see a metastasis that's new, or you could tell based on the previous images exactly what parts of the tumor are shrinking or growing, that is trying to tell some of that internal structure apart. And you're probably also going to use information about that patient's history. And all of this all together will go into an updated diagnosis. So in short, medicine is complicated, and it involves lots of different steps, all of which involve different aspects of medical image processing. Now, what if a patient walks into the hospital and their doctor isn't a human, but a robot? That is, what if they walk into the hospital and they're not being treated by a collection of humans, but they're being treated by an artificial intelligence? Well, the process is probably going to be very similar. They're probably going to get the same images, which means the same signals are going to be acquired. Those signals will probably still need to be reconstructed into a CT, and we'll probably still need to do aspects of identifying those tumors, segmenting them, registering with previous images, etc. So all of that pipeline that I showed you before can still apply. The only difference is we may be finding a machine intelligence to perform to perform those individual steps. Now, that's not the only way that we can solve these sorts of problems. If you've looked at machine learning in the news, you'll probably see people come up with these grand architectures that can do a lot of different things that seem almost impossible, where they can take a single image and from that magically come up with a diagnosis. And that is a valid approach. And that is a valid approach, and that can give you some very interesting insights. But what we need to consider is how we address this problem as a whole. So when you're making machine learning algorithms or any medical image processing algorithm, you tend to follow a fairly specific pipeline. That is, you tend to define what the problem you want to solve is, find an architecture to actually solve that problem, and then you train that architecture, you evaluate the models that you generate, and then yay, you publish. The important thing to take into account is that defining the problem to solve, as well as what architectures you use to solve it, largely depends on how you interpret what the problem is. That is, if you could take this very high-level view where your problem to solve is simply, here's a collection of X-ray images, tell me whether or not this patient's tumor is in remission or is growing. Or you could take the approach that we mentioned before, where you take your clinical problem, that is asking based on these X-ray images, is the tumor in remission, and breaking that up into those series of subproblems that can actually help you address that, that can help you understand and address that problem as a whole, and then finding different architectures to solve each of those different problems. Now, when you do that, another thing to take into account is this also changes what sort of data that you need. Because whenever you're training a machine learning algorithm, specifically the supervised machine learning algorithms that we see all the time in the literature, we need to make sure that if we break our problem into multiple parts, that we do have the annotations to support each and every one of those intermediate steps. So this defines what I think of as the primary axis in machine learning that no one talks about. So I'm sure you'll hear over the course of this program, you'll hear stuff about supervised learning and unsupervised learning and how there's the spectrum between them. And you'll hear about a whole ton of other design criteria, but I think this one's probably the most basic. And this criterion is simply how much do we separate the problem into multiple parts, that is what we call separable learning, or how much we treat the problem as a single monolithic whole, which in the literature and in the news is called end-to-end learning. So you can really think about it in terms of separable learning actually liking and encouraging intermediate steps, and those intermediate steps being something that you as a human define and understand. Whereas in end-to-end learning, you may have intermediate steps, but they're going to be hidden. And they're going to be something that the computer only has access to, and possibly the computer only understands. From the point of view of a clinician or of a designer, you really only get to see the inputs and the outputs. So let's take an example to make this a lot more concrete. So let's consider segmenting some of the small anatomy within the brain. So as you can see in this image, there's a lot going on in a very small area in the subcortical regions of the brain. You have lots of different anatomical regions that could be very interesting for a variety of different reasons. At my center, we perform deep brain stimulation. So in deep brain stimulation, there are certain regions of the brain that we're very interested in, and we want to be able to delineate very accurately because we're actually going to place an electrode right beside or inside them. So if you're taking an end-to-end approach, it's relatively simple conceptually, and people have done it. So you just take your image, you have some segmentation, convolutional neural network, probably based off of a UNet, who knows, and you simply put your image into that network. And at the and tell the network to output for every pixel what part of the anatomy that pixel represents. That is, at any individual pixel, you're asking the machine learning algorithm to tell me, oh, is that in the substantia nigra, or is that in the subthalamic nucleus, or is that in one of the mammillary bodies, or is that just in an area that we're not interested in at all? Again, this is a valid approach, and it has worked. It has been published, but there are complications. The biggest complication is actually that the structures that we're interested in are very, very small. They make up a fraction of a percent of the image. So this leads to a problem in machine learning known as segmentation class imbalance. So this is a generalization of class imbalance more generally. So class imbalance is when you have these labels that are so improbable, it's best for the machine to pretend that they never happen at all. So think of it as, imagine you're going to a city, a large city full of people, and a lot of people in that city play the lottery. Now, if you go to an individual person and you ask, what is the probability that this individual person won the lottery? The answer is pretty close to zero. And in fact, a machine learning algorithm is going to repeatedly just predict you did not win the lottery. So it'll go to Bob. According to our algorithm, Bob didn't win the lottery. Susan didn't win the lottery. Claire didn't win the lottery, et cetera, et cetera. And we'll go through every single person in that city and we'll say, for each and every one of them, you did not win. I predict that you did not win the lottery. Now, if we read in the news that someone in that city has won the lottery, but the news does not tell us who, then we have an interesting problem, because for our algorithm, it's still going to go through and predict that no one in the city won the lottery, that the probability of any of them winning is so small, it's negligible. But at the end of the day, that means our machine learning algorithm has said no one in the city has won the lottery, even though you have explicitly told it that someone has. Well, that's the issue with segmentation class imbalance. So we know that at least some of the pixels in this image belong to the subthalamic nucleus, because everyone has one. So even though each individual voxel has a negligible probability of being in the STN, we know we have to find some way of telling our algorithm not to just say the STN doesn't exist at all. So one of the approaches is to start treating this as a more separable problem and start thinking of other things that we could learn that don't involve classification and thus avoid class imbalance. One of the big things we could learn is simply the centroid of the region that we're interested in segmenting. This is not a classification problem. This is a regression problem. You take an image and you regress where in that image, what is the X, Y, and Z value of this particular thing. That is almost the definition of a regression problem, which means that you don't experience class imbalance. So one framework that Militari came up with, which I think is really interesting, is try to do that regression problem for each individual pixel. So that what that means is that it is now okay for the algorithm to overestimate the probability of any individual pixel being in the region of interest. That's like saying it's okay to massively overestimate our prediction of who wins the lottery, just in order for us to generate a list of people who could have won. Then we can use that additional centroid information to filter out the people who we don't think have, who we separate out the pixels in the image that don't make a sort of coherent idea of what that anatomy is. So that could be like separating out the list of people who might have won the lottery based on other supplementary information, like whether or not they bought a ticket. And we can even take this even further. So instead of predicting the centroid for every for every voxel and then determining if they're coherent or not, we can just literally take the entire image and just predict centroids. So when we do that, we're even giving the we're giving the framework even more information. Before, we weren't telling it how many of these objects it's looking for, thus allowing it to come up with as many as it wants. Now we're telling it exactly what object to look for and telling it precisely that there is only one. Now, what's the benefit of this? The benefit of this is we can do this first, and this can be done relatively quickly, much more quickly than segmenting the entire image. That is a benefit because once we know approximately where that anatomy is, we know that that anatomy is small. So we can take our entire image and we can crop it aggressively to that one tiny region where that anatomy happens to exist. Now we don't have a segmentation class imbalance problem anymore because the thing that we're interested in segmenting now takes up a large fraction of the image that we're segmenting, because the image that we're segmenting is now so small and is centered on the object that we're interested in. The other benefit of something like this is that we can correct this information if need be. And this is an important consideration in the clinic, because when you're trying to get algorithms that are actually going to be used in a medical context, we always have to be cognizant of the fact that these things can fail. And we have to be able to regulate them in terms of their failure cases. So if an algorithm fails, we have to think, okay, can we create something that makes it easy for a clinician to recover from this, as opposed to just have to close down the computer and start again from scratch and ignore the AI entirely. So I actually have a little demo of this system as it works, and it demonstrates some of the advantages of using separable learning. So one of the big advantages is speed. So just right now, as I was talking, it solved the entire segmentation problem. So the other frameworks would take minutes in order to process a single image. What you saw happens in seconds, and most of that was spent actually localizing the region of interest. So these are our two subthalamic nuclei that we're interested in, and it has automatically segmented them, and actually with a relatively high accuracy on the order of about 1 millimeter, which is the resolution of this image as a whole. But we could imagine that it may not identify this region at all. We could imagine a case in which one of the subthalamic nuclei is simply missing. Well, we could correct in this framework, we could correct for those types of errors. So a clinician could just sort of look through the image and say, oh, that's weird. One of my STNs is missing. It should be approximately here. Click on the image and tell the, here we go. There we go. Click on the image approximately where the STN is, thus telling the computer, hey, your localization was wrong, but here is a localization that you can use. Now run the segmentation. Part on the cropped image. So by separating the algorithm into multiple pieces, some of those pieces can be swapped in and out for different things. And one of those things is an actual human user. So that means if that piece happens to have an error, you can simply swap it out with your clinician. And if those pieces happen to do something that's relatively easy for a clinician to handle, like click on an image, then it can be done sort of quickly, and then it can be corrected quickly and efficiently. So we've looked at 3 different publications that all try to address the same problem. That is, they all try to segment this specific subcortical anatomy in the brain from an MRR image. On one side, we had this really sort of separable framework that I designed, where we have completely separated the problem of finding the centroid of the object that we're interested in and segmenting it. Then we have the framework that was built by Militari, the one where for every pixel, they're trying to estimate the centroid and then determine what things are coherent. So in that framework, it's a little less separated. We have a single framework that tries to get the centroids and the segmentation, but both of them at the same time. So it's a little bit separate because we interpret them differently. And we process them differently afterwards, but it's at the same time, they're still learnt at the same time. So it still has that end-to-end flavor to it. And then finally, the first framework that we looked at, where it just immediately went into a convolutional neural network that segments it directly, is a very canonically end-to-end way of treating the problem. We have no idea of centroids. We have no idea of how big these structures are or where they are or how numerous they are. We're hoping that the computer can learn all of those by itself from the data. So my first piece of advice to you is to, for whatever problem that you want to solve, is first to think big and start with the big picture. And I mean this in two different ways. So you can start with the big picture in the sort of end-to-end way where you think, okay, what is the input to your problem and what is the output? That is a very big picture way of thinking about a medical imaging problem. So you could think of something like, oh, okay, my input is retinal images, so images of the eye, and my output is a diagnosis of if this person has some sort of eye disorder like retinal degeneration. So that's a very big picture way of viewing the problem. But I want you to also think about the big picture in terms of how it's done in clinic, how a human performs that problem right now. So that means looking into the literature, not only in machine learning, but also looking into the literature in medicine to see how are people addressing that exact problem right now. What are the steps that they're using, and what is the meaning, what is the purpose behind any individual step? Now, for some of this, you may even need to observe an actual clinician. You may want to engage in some sort of ethnographic research where you watch a clinician in the wild, where they're trying to, say, segment the SDN, and you'll find that what they'll do is they'll try to approximate where in the image that SDN happens to be, and then they'll zoom in on that and then segment the image. Well, that was the motivation behind how I created that separable framework, is I literally watched people as they try to perform this problem and then try to create an artificial intelligence that can mimic all of those individual steps. The next thing that you want to think of in terms of a big picture is what the patient population is like and what assumptions are safe to make. So this is important because the steps that people go through may change based on things that they recognize or realize about the patient. So if a patient, say, comes into the hospital and they're complaining that they have a headache, one of the first things that we'll do is we'll take a CT image. And that is to check to see if that person is having a stroke. Now, if they are having a stroke, then we want to be able to segment or determine where that stroke happens, where that stroke is occurring. But if they're not having a stroke, we will want to do something different. We may want to see if there's some sort of abnormality. Maybe we can see a tumor. Maybe we don't see an abnormality and we'll want to schedule some other type of imaging. So knowing what the patient population is like and knowing what their distribution is can tell you, in terms of what steps you're using, what are safe approaches and what are approaches that we'll have to keep in mind in terms of actually translating these into a complicated clinical situation. So what do? Do you choose a more separable architecture or a more end-to-end one, based on an understanding of that problem and those steps? Well, it depends on what your goals are and on what else you have available. So one of the and the rest of a lot of this presentation is going to be structured in terms of comparing the benefits and the disadvantages of these two different ways of thinking about and addressing these problems. One of the distinct benefits of having your problem separated into different parts is it's a lot easier to compare things. And that is because, as I mentioned before, you can take different parts of the algorithm out and replace it with other things. So that means you can take parts of your algorithm out and replace them with, say, something from the literature. And then you can measure how well this new overall framework works. And from that, you can tell how important or how performant that individual piece that you designed and took out happened to be. That's called an ablation study. So when you have separable learning, it turns out that it's a lot easier to perform these sorts of ablation studies. And that gives a lot of credibility, not only to your research, but also to the publication that, that your research will eventually become. It also means that that your algorithms will become easier to reproduce. And that is because no one has to reproduce the entire thing. They can take individual parts and reproduce it on different data, possibly for a different problem. And ultimately, that means that when you're building a framework using separable learning, it's ultimately safer because you're focusing more on this sort of incremental progress. Now, on the other side, if you want to do something end-to-end, you have a lot of novelty there. You have the opportunity to disrupt the way that people are doing things currently. And that could be a good thing. You'll find that a lot of clinical workflow is determined by best practices that were written about in the 70s or in the 80s, long before any of us were born. And that may not be the best way to perform these tasks. There's a lot of inertia in the medical community in terms of how to perform certain tasks. And by using end-to-end learning and by using the sort of perception of novelty, you can break out of those ruts and maybe find something that's really new and high performant. But that also makes it a lot riskier. There's a lot more variability in terms of how to do something new, as opposed to how to do something the way that people have been doing it before. Now, a lot of what I just talked about hinges on the idea of reuse. So one of the reasons why separable learning has this incremental progress and has this sort of safety to it is because it focuses on modularity and explicit reuse. That means that what I mean by explicit reuse is you can take a part of the algorithm that you know about and then literally explicitly use it for something else. So for my STN segmentation framework, for example, I could take the part that localizes the STN and determines where it is, and instead of using that to segment the image, I could use it in something that helps me, say, register an image. Maybe I have a bunch of these localization algorithms, and then I can literally match points across two different patients and then register two different patients together based on those points. Well, now I've explicitly used part of my algorithm for something completely different, simply because that problem, even though it's different, even though it may have sort of different inputs and different outputs and maybe used for an entirely different thing, the fact that it could share a sub-problem means that you can reuse some of these algorithms. Now, end-to-end learning can still be reused, but the type of reuse is now a little bit more nuanced and a little bit more complicated and a lot more implicit. That is, we're not going to be explicitly taking one of our boxes and then exactly using it, parameters and all. Instead, we're going to be taking it, tweaking it a little bit, or taking inspiration from it. You'll see a lot of interest in things like transfer learning. That is when we literally take a part of the model and instead of explicitly reusing it exactly the way it is, we use it to initialize our training. That is, we use it, we say that, hey, this model possibly is close, but needs to be tweaked. And then we perform an additional training process in order to change that model to make it work for this new problem. So that's implicit because yes, we are reusing it, but we're not reusing it exactly the way that it was. Similarly, we could be reusing different knowledge from these machine learning papers. So what you'll find for, say, a segmentation problem is lots of people are using UNets, and they'll use not the same parameters, not the same weights, but they'll use the same structure. They'll use the same number of layers, maybe the same width of all of those layers. And that is another example of implicit reuse. That is, these algorithms are designed to be so flexible that you can take the general description of them and use them for different problems, provided that you have the training data available. The next thing you should consider is your data. So this is probably the most important point in terms of separating, in terms of understanding the difference between separable learning and end-to-end learning. Separable learning requires a lot more attention to the data. That is, for any individual data point, you're going to need to generate a lot more information. So let's take our cancer example. Imagine so the problem is someone comes in, you take a CT, and you want to know cancer or no cancer. Or let's make it even better, malignant cancer, cancer that we have to treat, or a benign cancer or no cancer at all. Now, if we're going to use separable learning, well, then we'll probably need a lot of annotations. We'll probably need something like, okay, does this person have a tumor or a lung nodule? Yes or no? Where are those nodules located? Can you segment those nodules? What are measurements of those nodules that can help you determine whether or not they're cancerous or non-cancerous? All of that additional information is going to need to be added into your annotations. That is because when you're training a separable learning model, you're going to have to be training all of those intermediate steps as well, which means you're going to need desired input and output for all of those intermediate steps. So that means that you're going to need a lot more information about each and every individual patient. But there is a small benefit. And that is that separable learning algorithms tend to be a lot more efficient with the number of patients. Now, this is because you're providing a lot more information about each individual patient. So it kind of makes sense that when you're providing so much information about that individual patient, you don't need as many of them. Now, over on the opposite side, end-to-end learning is the exact opposite. Because you're only focused on the input and the output, you don't need to annotate all of those intermediate steps, because you're leaving it to the computer to try to invent its own intermediate steps. So you're not going to tell it, okay, at this point, you're going to need to estimate the location of a nodule. No, you're going to trust it to figure that stuff out by itself. All you need in terms of data is the input at the very beginning, like the CT scan, and the output at the very end. That is whether or not the cancer is malignant, or if it's benign, or if they don't have cancer at all. Now, this ease of annotation is great because it means that we can get a lot of that information from things like hospital packs and hospital systems that exist already. And this is because in clinical routine, clinicians currently throw out a lot of the work that they do. That is, they'll perform, and they may not do this consciously, but they'll perform certain tasks, but they won't save the information from those tasks. So you may see a radiologist scroll through an image, find a tumor or find a nodule and think, hey, that's a nodule here. But then instead of saying, oh, there's a nodule at this particular XYZ coordinate, they'll just say nodule present right lung. So that information will make it into the packs, and it'll be easy to extract, but it won't be as informative. This means that the data that you get is simply larger. Larger data sets are available to you simply because you can start using these hospital pack systems without asking the clinician to add more and more annotations, which they may not have time to do. Now, there's one other sort of side benefit of having separable learning and having this data separated and treating the problems as separate problems, and that is distributed training. This is really important because machine learning algorithms can be very memory intensive, they can be very large, they can take a lot of time. And if you have multiple computers, you may want to actually have different computers process or train different parts of your algorithm. Now, if you're using separable learning, that works no problem. You can have computer one train algorithm one, computer two trains algorithm two, computer three trains algorithm three. If you have end-to-end learning, it's a lot more difficult because you only have one algorithm start to finish. Now, you can distribute some of the tasks, like you can say, okay, I'm going to give some of the patients to computer one, some of the patients on computer two, some of the patients on computer three, but you're always going to have additional work put in terms of reconciling the models that are learned by each of those different computers. So that is called federated learning. Again, you'll probably hear a lot about it. And that is a way to distribute the training for end-to-end models. But the key thing to understand about federated learning is it's a little bit of a hack. It requires additional communication between the computers that you're using. It requires more complicated procedures for aggregating or combining models. And all of this is because end-to-end learning fundamentally is not designed for distributed training in the same way that stuff like separable learning is. So you'll find that with separable learning, you probably won't need a second computer. You probably won't need something like federated learning in order to make these algorithms work. You can probably do it at a single computer in a single clinical center and still get high performance results. So, second point, think about your data. So think about where this data is coming from. If you're collaborating with doctors, do you have access to their PACS system? Do you have access? How much time do you have access to? Can you build things that will make their life easier and give you those additional annotations that you need to perform your actual research? The other thing that you can consider is collaborating with other researchers and seeing what data they have available. Because especially if they're interested in a slightly different problem than you, then they may have some of those intermediate steps already, or they may be developing, or they may themselves be developing algorithms to find those intermediate steps that you can then take advantage of. The other thing that I strongly suggest, especially if you're doing end-to-end learning, is to look at open databases. Here's just a small collection of the many, many, many open databases there are in neuroimaging. We have some that are specific to a given disease, like the PPMI, which is specific to Parkinson's disease, or ADNI, which is specific for Alzheimer's and cognitive impairment. You may find ones that are more structural, like OASIS or the Internet Brain Segmentation Repository. Those aren't specific to a disease, but they're trying to address a more general problem that can be used for a lot of different diseases or a lot of different procedures, like segmenting the image or parcellating the cortex into different parts. You may find things that are longitudinal, like the Infant Brain Atlas or many of the databases in brain code, where you're looking at how brains evolve over time, which could be used for different types of algorithms. You may want to design things that sort of predict how the brain develops with time from earlier time points. And again, you can use different frameworks in order to perform that sort of prediction, or in order to make use of those sorts of predictions. The last thing that you could always do is you could always make the data yourself. This is particularly important for open databases. So don't be afraid to take an open database and then to annotate it with additional information that you would want or that you would need to solve a particular problem. You could take an, so this also involves, so this could be, if you're doing some, dermatology is a great example of this, because you can literally talk to the other people in your lab and get photographs of, different moles or different marks on them, and literally generate your own database of healthy volunteers, which you can then use to supplement some of these other databases or extend them in terms of the demographics of their patient population. Never be afraid to literally make the data yourself and to provide additional annotations that you think you need. The one thing that you should always consider, though, even when you're making the data yourself, is to balance the informativeness of the annotations. That is how much information you give for each and every patient, and balance that off with the ease of annotation. That is how easy it is to collect this data, and thus how many patients you can collect this data from. Another piece of advice, and this is one that I give all of my students when they just start performing their first study, and that is to ask the question, are you science or are you technology? And I don't mean this in a normative way. Both are good, both are necessary. I'm just asking you, why are you making this particular artificial intelligence? Are you making it to further science, or are you making it to further technology? And these are two very different problems with very different considerations. If you are technology focused, maybe you're trying to diagnose a disease, maybe you're trying to create something that's used in clinical workflow, then you should focus on things like accuracy, usability, the ability to correct or detect errors, the ability to fall back to different systems. Those are all things that we have to consider in clinic. And we have to always ask ourselves, what is the added value that we're getting from incorporating artificial intelligence? If we're doing science, then instead of trying to just diagnose a disease, we're trying to better understand it. So this means that we don't necessarily have to focus on accuracy. We don't need something to diagnose a disease with 100% accuracy in order for it to be scientifically useful. It just needs to be better than chance, but explainable. That means that it doesn't matter that we have 99% accuracy. We could have something like 70% accuracy, but our framework tells us exactly what we're using in order to get that accuracy. And because we know exactly what the computer is looking at, we can then intuit that what the computer is looking at is correlated with that actual disease and furnish hypotheses from that. So when you're creating something and you want it to be focused on science, don't focus on accuracy. Instead, focus on explainability. Don't focus on value, focus on knowledge. So the thing about machine learning and artificial intelligence is it is very good at generating information. But information is not knowledge. I can give you a random collection of phone numbers from the phone book here in France, and you will have no idea what to do with it. I've given you a lot of information, but I've given you absolutely no knowledge. Whereas if I give you, say, my cell phone number, and I say, this is my cell phone number, I've given you relatively little information. I've only given you a single number, but I've also given you the knowledge of what you can do with it. You can phone me. If you want to ask questions about this presentation, well, here's my phone number, just phone me. Now, there is a really big difference between separable and end-to-end learning here, and in terms of how we move from information to knowledge. And it all has to do with this philosophical concept called logocentrism. So logocentrism is actually it's almost an entire century old. It's a 100-year-old idea from philosophy. And the idea behind logocentrism is that complex knowledge is expressed as text. Fundamentally, we understand the world through text, through words. And this is a very sort of powerful and a very important idea. And you can trace the roots of it all the way back to thinkers like Plato. When Plato, if you know anything about Plato, then you know about his theory of forms and the idea that, say, every tree individually takes inspiration from the form of a tree, the perfect tree, our mental conceptualization of what a tree is. But the thing is, knowing or having the ability to say the form of a tree requires us to have a word for tree. We don't have forms of things that we don't have words for. The word itself is fundamental for us to be able to create concepts and to group all of those concepts together. Now, this is a huge benefit for separable learning because we ourselves are fundamentally bringing logocentrism into the problem with us. When we create intermediate steps, we are creating them as humans who think in this logocentric way. So we fundamentally create explanatory objects. That is, we fundamentally create objects that we have words attached to. And we have ways of, we have words that tell us how these things are related. So if we're, say, trying to do this tumor diagnosis problem, and we're doing things like measuring the size of the tumor, well, I'm fundamentally bringing in concepts like tumor. I'm fundamentally bringing in concepts like volume. And these are concepts that we have words for. And this is great because if we have words for them, we can literally write those words down in a publication and submit it to a journal. If you don't have the words, it's a lot more difficult to do that. And that makes it a lot more difficult for your machine learning algorithm to go further. Now, end-to-end learning is different because we are no longer bringing logocentrism in with us. It's the computer that is determining what the intermediate steps are. And a computer does not think in a logocentric way. Computers fundamentally don't think in words and language the way that we do. They think in terms of numbers and computations performed with numbers. And it never has to explain what those computations are. Explainability is something that we have to bring in. It's something that we have to then impose upon the algorithm, either in terms of imposing it on the structure, the same way we do with separable learning, although with a lighter hand, or imposing it on the structure afterwards in terms of creating explainability frameworks like attention models, like a GradCam, that then take this non-logocentric model and try to explain what's going on inside it in a way that we can see and in a way that we can then translate into words. And this is because when we're creating end-to-end learning models, the purpose isn't to explain a solution. The purpose is to just solve the problem. It doesn't matter what the solution is. It doesn't matter if you can explain it or not. All the algorithm knows is that here is the structure of the solution, find the best one. So let's get some more concrete examples. So here is a separable learning framework that I helped build half a decade ago that diagnoses temporal lobe epilepsy from three different brain images, a T1 weighted image, a T2 weighted image, and a DTI image. So what we do is we literally say, okay, I'm interested in these particular regions that I have names for, like the anterior pole of the temporal lobe. That is a well-defined region that we have a name for and that we can measure. So we can then measure what is the intensity of the T1 image in that area, what is the intensity of the T2 image in that area, what are the diffusion characteristics from the DTI in that area. So now we have measurements that we can explain of those areas that we have words for. And from that, we can then use some form of simple classifier, like in this case, it was a linear SVM, to separate our patients from our controls. At the end of the day, what do we have? Well, we have a vector from our SVM, and that vector actually gives us a lot of information. It tells us what parts of the image that we're actually interested in. If our SVM vector has a 0 in for, say, the T1 value of the anterior pole of the temporal lobe, well, then we know that the anterior pole of the temporal lobe, at least in terms of its T1 intensity, does not help us solve the problem. It's not related to to epilepsy in general. That's just an example. It probably is. I don't know off the top of my head, but you can see how we can take those specific numeric values and assign a meaning to them really, really quickly, simply because it's a nice, simple linear model. Linear models are great because, as I showed, they allow for this very easy explanation. Numbers that are, if we have big numbers that are positive, well, it's because those features that it's looking at are strongly positively correlated. But there's other benefits as well. One of the main benefits of these models is we can do things like create confidence intervals and do statistical testing. That means we can tell how certain we are that the conclusions that we're making in our paper are correct, that they're really based on the data that we have and aren't just some sort of statistical fluke. The other benefit of linear models is it's a lot easier to create counterfactuals and guide predictions. We can ask questions like, okay, we have this patient. It says that they don't have temporal lobe epilepsy. I wonder what it would look like, what this patient would look like if they had deterioration in the hippocampus. Well, you can model that easily. You take whatever your measurement is for deterioration, wherever the hippocampus is, and you can just say, okay, if I change that value, well, now I can look in my big SVM vector to see, okay, if I change that the value of that degradation by this much, well, that's multiplied by this number, that will change my prediction by this much. That makes it very easy to create these counterfactuals and to ask questions like, what if? The other, again, very practical benefit of linear models is they're acceptable almost everywhere. Every journal is going to be perfectly okay with linear regression if you use it properly. You don't have to explain it. You don't have to justify it. You don't have to be like, oh, we're using linear regression because we actually believe this relationship to be linear. It doesn't have to be linear. You can still use linear regression and your paper will probably still be accepted, simply because these methods are so widely used and widely accepted. They may not be the most attractive in terms of machine learning models, but they're still the most popular for a very good reason. Now, here's an example of an end-to-end style framework that I created in my lab last year, where we literally had to build in an explanation framework in order to extract knowledge from it. So it's a very traditional end-to-end architecture. We have DTI images, and we're predicting whether or not the patient has, in this case, Parkinson's disease. So all we care about is images, Parkinson's disease. We're not going to segment anything. We're not going to localize anything. We don't care. We're not even going to do brain masking. We're just, all we have is image, disease, go. So the algorithm performed at about 70%. So it's not, generally speaking, usable in clinic. We can get much higher accuracies by simply doing things like asking the patient to perform simple tasks and measuring their performance on those tasks, like writing their own name. And from that, you can get a more than 70% accuracy, a more than 70% chance of determining whether or not they have Parkinson's disease, simply from looking how much their hand shakes when they write their own name. So clinically, not terribly useful, but we want to use this for science. We want to use this sort of framework to tell us something new about Parkinson's disease. So what we did is we developed our own explanation framework. We used backpropagation, which I'm sure you've either heard about or will soon hear about, to get the derivative of our prediction with respect to each and every voxel in the image. So these are sensitivity maps. This essentially correlates with attention. So regions of the image that have a high sensitivity are regions of the image where the computer is actively looking. Now, this means that for each patient, we have different regions of the image that could be highlighted. So again, on an individual patient-by-patient basis, this is not terribly useful. We can still get more clinically accurate, more relevant algorithms. But what would be interesting is if we can group all of our patients together and see maybe some of the and see how robust some of these signals are. Maybe all patients sort of perform the same way. Maybe some patients perform different, some patients have different sensitivity maps than others. So one easy way to do this population-based analysis is through registering all of these patients to an atlas, like the Montreal Neurological Institute atlas, and then using voxel-based analysis to tell if any of these sensitivity metrics are consistently positive or consistently negative. That is, consistently telling our computer that, yes, this area is interesting and is correlated with Parkinson's disease, or hey, this area here is interesting, but it's because it's negatively correlated with Parkinson's disease. So if we separate Parkinson's patients from healthy controls, we ended up getting maps like this, which gave us really interesting results. Now, we all know that Parkinson's disease affects the basal ganglia. So the fact that it's highlighting things like the globus pallidus and turnus is something that we already know. What we didn't know ahead of time is that cerebellar involvement is a lot more distinctive than basal ganglia involvement when it comes to diffusion tensor imaging. That means that, funnily enough, if you want to diagnose Parkinson's disease, you may not want to just look at the basal ganglia, even though we know that's where Parkinson's disease originates. What you may want to look at instead are the fibers that are attributed to motor symptoms, because the changes in those fibers actually overwhelm the original upstream changes that you saw in the basal ganglia, simply because they're bigger, they're clearer, they're easier to see, and thus easier for us to diagnose with. So this gives us a very good, very strong scientific hypothesis that tells us something. We've now learned something about Parkinson's disease from an end-to-end model, but we had to build that explainability framework on top of it. There we go. Oh, I'm going the wrong way. There we go. So without some form of guidance, without some form of explainability framework, interpretation can be difficult. And even when you have an explainability framework, interpretation can still be difficult. And this is because, as you saw in the previous example, you can see multiple things at the same time. In fact, you could be seeing everything everywhere all at once. So we don't, from that previous image, I don't know if there's some sort of correlation between the regions that we've highlighted in the cerebellum and the regions in the basal ganglia. Maybe some patients have this really strong signal in the cerebellum and others have a really strong signal in the basal ganglia. Maybe the patients both have this strong signal in both locations at the same time together. I can't tell that apart based on this explainability framework. This can be very disappointing sometimes. So we applied this exact same framework to a different clinical problem. We chose frontal temporal lobe dementia, which is dementia caused by the degradation of the frontal and temporal lobes. And guess what our explainability framework told us? It literally just highlighted the frontal lobe and the temporal lobe. And yeah, duh. We haven't learned anything simply because it gave us everything everywhere all at once. Yet we already know that frontal temporal lobe dementia is caused by degeneration of the frontal and temporal lobes. We don't need a machine learning algorithm to tell us that. So this is, even though it's a success in terms of creating an algorithm that has learned something for itself about frontal temporal lobe dementia. In fact, it learned that it happens in the frontal temporal lobe. It hasn't taught us anything, because it's still showing us everything everywhere all at once. We need to be more precise. So you may not be able to see anything useful unless you ask the right kinds of questions and train the right kinds of end-to-end machine learning models. So for example, if instead of asking the model to differentiate between healthy controls who have no history of Parkinson's disease and people with Parkinson's disease, maybe we can ask a question like, hey, can you tell people who were just diagnosed with Parkinson's disease who have very, very minor cases apart from people who've had Parkinson's disease for 5, 10 years and are so shaky that they can't write their own names. Well, that's a more difficult problem, but it can give us information that can be a lot more sensitive. So when we asked the computer to distinguish between these two groups, we actually got information that's a lot more specific. In fact, it told us not to look at the onset side, the side that Parkinson's disease starts on. It tells us actually to look at the opposite side. And this is because as patients move from this mild disorder to something more debilitating, what's happening is the degeneration is actually moving from one side of the brain and spreading to the other side. So this is something that we already knew happened, but our machine learning algorithm is now telling us exactly where it happens. It happens in the globus pallidus internus. That is the part that gives us the most, the strongest signal for distinguishing between these two groups of patients. We have now learned that that globus pallidus involvement is what really defines the progression of Parkinson's disease, not motor symptoms in the cerebellum, not degeneration of the substantia nigra, but this region right here. In terms of frontal temporal lobe dementia, instead of asking it just to, say or distinguish between healthy controls and patients with dementia, instead we can say, okay, let's do a regression problem for all of our patients with dementia. Let's see how well we can create an algorithm to say predict their capability to recognize emotions. So that's something that degrades with dementia as your dementia case gets more and more severe. You become less and less capable of seeing emotions. Let's find out where in the brain that's happening. And by doing that, our algorithm has actually learned a very precise region in the posterior temporal lobe that tells us exactly where the degeneration is happening that corresponds with this particular symptom. That means that we can now tell apart different types of dementia patients. Patients that have dementia where there's this emotional involvement, it's because of a degeneration in this posterior part of the anterior temporal lobe. If they don't have this emotional recognition deficit, it's because they don't have this type of deterioration in that particular region. They are deteriorating elsewhere, which would explain their other different symptoms. My last piece of advice to you is to do something fun. Do something that you are passionate about. All of the examples that I've shown you are something that I find deeply fascinating. And that is something that you'll want to look for in a project. Find a project that you love, because if you love what you're doing, trust me, you're going to do it really well. I have faith in each and every one of you, and I know that you are going to define and solve interesting problems. So I would like to thank, firstly, Islam for inviting me to give this talk, for having faith in me that this is something that that can help in this program and can help you develop as AI researchers. I would also like to thank one of my colleagues, Pierre Janin, for some really, really fruitful philosophical conversations about how machine learning works and about how it's starting to affect how we perform digital computerized medicine. Lastly, thank you all for your time and your attention.
Thank you very much, John. Thank you for this fantastic and transformative talk. You brought, like so many important points. I'm sure that the audience, I'm sure that they have a lot of questions now, even if, we have some limited time to just take a few questions. So maybe we can start with Cinda. Cinda, would you like to ask your question directly? She raised her hand.
Yes,
and
please.
guys, feel free to turn the cameras on if you want. Yeah.
Sure. Hello.
Hi.
Hi. So first, thanks a lot for your time, and thanks a lot for this wonderful presentation. We sure did learn a lot. So my question is, earlier you've mentioned the difference between end-to-end learning and separate learning. So from what I've understood, the steps to follow are building the model definitely after addressing the problem and building our hypothesis, etc., etc. So building a model, train it, test it, then we perceive the result. If they're accurate, then great, wonderful, yay. If not, we point at the missing information that perhaps our model has missed or a mistake that it has committed, then we correct this mistake or missing information and redo the whole process. So my question is, what makes end-to-end learning different from transductive learning? So aren't the two approaches quite similar? Is it the fact that in end-to-end learning, we follow a certain pipeline, let's say, training, then testing, which is not the case for transductive learning, because we do the whole thing as a whole, train, test, all together, loss minimizing, and redo the whole process until sufficient results are reached. So
they're really orthogonal. So you can use transductive learning and separable learning at the same time. So you can use that sort of transductive learning framework and apply it to each individual multiple AI models that you're using to solve a problem in a separable way. So they're really orthogonal concepts. Separable and end-to-end is not about how we train models, it's about how we solve problems. So anything regarding training, like supervised versus unsupervised or transductive versus traditional, you can use at the same time as this as separable learning or end-to-end learning. They're really orthogonal concepts.
Great, thanks.
Great, thank you very much for clarifying, John. Sufyan, you have a question? Please go ahead.
Yes, thank you very much. Thank you for this rich information. In fact, many ideas are coming at the same time. Separable learning. Firstly, can we say that if we can learn from a piece of data, so we can save privacy? Is it true? Like federated learning, we can just exchange or learn from a piece of data, not whole data. So we can save privacy in that case.
Privacy is a lot easier to ensure with separable learning. And this is because you can take the parts of the data that may be private and may be confidential and train models that only deal with those particular parts of the data in a safe, private, and secured way. And then other parts of the data that can be more public can then be made public and can be trained in a way that doesn't have to preserve privacy. So for example, you may have something like a list of tumors and a list of various measurements of those tumors, and you may want to correlate whether those measurements with whether or not that particular tumor is malignant or benign. You have no identifying information there. So that means you don't have to care about privacy for that particular task simply because you don't have any private information. And that is because the part that was private, going from the images to those tumor measurements, has been separated and has been dealt with in a private location.
It can stay anonymous, man.
Exactly.
Okay. Second thing about edge AI. Can we merge separable learning? In fact, if we make analogy, it's like oriented programming approach. We segment the problem into sub-problems, sub-modules that we call objects that we can reuse.
Exactly.
We can link together. So if we can implement one object or one piece of this model or one sub-model into an edge AI, an edge board, or tiny, like tiny ML nowadays, for example, implementing the deploying the model on microcontroller or we can do this?
It's certainly a lot easier to do that. Yes, this is a side effect of distributed training because we're training smaller models on smaller models for smaller problems on smaller data sets. We can use stuff like edge computing and smaller devices.
Okay.
Again, if you want to solve a big problem, you may need many of these small models and thus many of these edge devices. So there is a scalability thing to consider there, but the whole key is your scalability is controllable.
Other thing, can we rely, is it reliable, this separable learning as compared to end-to-end learning? That's the question.
It's hard to say for sure. Right now, the answer is yes. Separable learning has been more reliable than end-to-end learning, but that might change. I'm not confident in saying that separable learning will always be more reliable. It just so happens that right now it is. Maybe in the future, we'll have data sets that are so gigantic and machine learning algorithms that are so powerful that it will legitimately be better for the machine to learn intermediate steps for itself. But right now, we have 100+ good years of medical literature that give us those intermediate steps in a way that's much better than right now we can train with data that we have available.
Okay, thank you very much.
Thank you very much. Sorry. Yeah, thank you, Sofiane, for the questions. And thanks, everybody. John, it has been a great delight to have you. I'm sorry, guys. We need to wrap up the meeting because I have another meeting to join in a minute. Sorry about that. But if you do have your questions, please write them down. You can email me the questions. I'll make sure to communicate them to John. And I know John will take some time to answer your questions and I will broadcast to all of you his answers, okay? I know you have your burning questions. Keep them on fire and he will just fire back. Thank you so much, John, again. And everybody here, and I hope I'll see you all soon. Thanks for the great talk.
Thank you for inviting me. And I can't wait to see what you guys do. This is going to be fun.